All right, thank you so much for coming out on this rainy day. Again, hopefully this is a little retreat from the wetness. I decided to bike from my house, so I'm happy to be inside right now. I'm Ashley Kidd, and I'm really excited to talk about this work that truly does embody hundreds of people, if not thousands, and the Sunflower Star Laboratory is we're just very happy to be part of the story of sunflower sea stars and how, especially in Monterey, and how we can help them. The Sunflower Star Laboratory, first of all, how many people have heard of the Sunflower Star Lab? Wow, okay. This is great. You guys can tell me what's going on then. So how many people have been to the Sunflower Star Laboratory? Okay, excellent. Yeah, Sunflower Star Laboratory, indeed. So we are a Monterey-based nonprofit in Moss Landing. If you were at the prior talk, they had a really great overview of the bay and this beautiful canyon that we live next to. And we're literally across the street from Umbari. So they're great neighbors. We love our neighbors and the community that are in Moss Landing. This is early photos from our lab in 2024, 2025. We were gifted this space and were able to move out of Vince Christian, who's up in the image there, who started it in his garage in Pebble Beach. So we're really excited to be in this space and be a community-centered organization open to the public. And yeah, we are 100% dedicated to sunflower sea stars and help collaborate and bring together other sunflower sea star researchers to help sunflower sea stars recovery and restoration efforts. So if you've already heard about our lab, and most of you have been there, you know maybe what they look like. But just so everyone is aware, sunflower sea stars are related to all these wonderful cousins on the side of the screen. So they're a sea star, but they're also related to urchins and basket stars and cucumbers and crinoids, these also beautiful animals that you would see in the Monterey Bay and worldwide. But our particular star of the show today is this sunflower sea star with multiple arms, up to 24. They don't have to have 24 arms. And actually what's very cool in this photo is, you can even see over here, there are two new arms being grown. Another interesting thing about the sea star is you wonder, how do they end up with all these multiple arms? And this is kind of a clue. They actually will grow arms at mostly two distinct parts, and then they just kind of keep growing around the star, and they keep adding them as they get bigger. So you can see those two little arms down there are the newest members of the arm family for this one individual. And this is also a beautiful picture. Again, they come in all sorts of colors, and you'll see a bunch of sunflower stars throughout this presentation. But this little button here is called the madreporite, and that is the pore that I can talk more about, but that brings in the ocean water and pretty much runs the whole species. So all these animals run on ocean water. They don't have blood or any other type of fluid. They basically just secrete what they need from their body into the ocean water that allows them to move, allows them to digest and respire. So all these animals have been on our planet Earth for 450 million years, so they've developed a pretty robust way of sticking around. Couple things that you may not see when you're just waltzing by a sea star is their way of digesting. So this is an upside down sea star. You can see they're beautiful tube feet. So Pycnopodia helianthoides is the scientific name for sunflower sea stars. And pycnopodia just means dense feet. They actually have more sets of tube feet that help them get around more than any other sea star. That's why their genus has that name, and they're the only species in their genus. And then that's their gut that they're everting, because they can digest animals from the outside or engulf them. Like their stomach can open up like an aperture, and they can basically just engulf something that's about the size of their central disc. And then this is sort of a close-up. You can see this is all the papillae and then rosettes of Pedicelaria. And the papillae are actually commonly known as skin gills. It's how they respire. They can diffuse. This is actually tissue that's coming out through their exoskeleton or endoskeleton, and it's just allowing exchange of diffusion of waste materials from metabolism and metabolic use. And then these little rosettes, they do look kind of, if I had a better picture just of these rosettes, it really does look like a bouquet of roses. But if you were a tiny little thing on the surface of a sea star, these rosettes actually have little pincers. And so they have a muscle and pincers, and so if you were ever at a touch tank in an aquarium and you had your arm in the water, you might have felt like a little pull on maybe your hairs of your skin. I don't know if anyone's felt that before, but that's actually these little pincers that are on the surface of stars. And it's not exclusively to this species. All different sea stars have like a different variation in the shape of these pincers, but when they first discovered them, 100 years ago, and started looking at them, they thought they were a different animal living on the surface of a sea star, because they were like, what are these little pincers going on? Anyways, fascinating. The other crazy thing about sea stars is they're highly regenerative. So this is an image of a beautiful, vibrant red sea star, and you can see the arm is huge. It's like almost coming off the image, and then a bunch of these little arms are regrowing. So not only do they grow new arms, but one of their defense mechanisms is that they can drop an arm and run away if they're being preyed upon. So this is quite an impressive image of a species or an animal that's been attacked maybe by a crab and had a bunch of its arms pulled off to get away. And that's the nice thing when you have up to 24 arms. You can keep moving on. And another interesting thing about sunflower sea stars and sea stars in general, is every single arm has the same organelles. So at the tip of every arm, so right here and right here, and up at the other side, and over here and here and here, they will all have little eye spots to sense their environment, light and dark. They'll all have reproductive organs in every arm, and they'll all have digestive organs in every arm. So if they drop one arm, they're not losing a one-of-a-kind thing. Again, it's redundancy within a single individual. This is a beautiful, this is the picture of the skin gills, the papillae, and those rosettes up close. They're actually, yeah, they're very fluffy, is what I like to call them. And so this is how they locomote. How do you coordinate 15,000 tube feet, which are those things moving along the bottom, when you don't have a brain or a front or a back? And a lot of it is just it's innate, they're all just like pressure sensors. They're kind of just navigating based on chemical cues and localized suction and decoupling from the ocean floor. So if you think of it, if you've seen an octopus or had an octopus encounter, they also have suctions. And at the end of each tube foot, I'll play it again. It looks, if you went up into the arm, it's like a turkey baster, where the top of the turkey baster is controlled to suction or release from a muscle movement, muscle contraction. And if you contract the muscle, it squirts the water out and releases the foot so it can move away. And when it wants to suck back in, the muscle relaxes and it can suck back onto the ground. And that's how they move across their environment, with all those 15,000 tube feet all doing that. Oh, now I can't get out of here. Okay. There's also a lot of multi-armed sea stars. So I was aware that there were multi-armed sea stars because I used to work with coral reefs, and I was aware of crown of thorns. I don't know if you've heard of crown of thorns in Australia that eat coral, but they're a multi-armed sea star that that I knew existed. But just in the eastern Pacific or off our coast of North America, these are some other species that you may come across, or in Antarctica. This is a very multi-armed sea star. This is Heliaster cubanigii, which is found in the Gulf of California, and looks very similar to a sunflower sea star, but this is the only sunflower sea star out of the ones on this image. So this is another sea star that is actually found, it's on exhibit in, at the Monterey Bay Aquarium. This is Rathbanaster californicus. This is a deep water sea star that you would actually see in a lot of those Mbari videos, but they're found really deep, like 1,000 feet deep, and they're only almost exclusively orange and red. Whereas sunflower sea stars, as we've already seen in the past few images, they can be purple and blue and red and all sorts of colors. And then you can also tell a sunflower sea star apart based on sort of the proportions of their central disk. So this is the central disk. And then sort of like the length of their arms. So I also had to learn really quickly identifying sea stars, because I was shown a cubanigii early on, and I was like, oh my gosh, they're all over the place, but they're not even in the same range as sunflower sea stars, and we'll get to where they are in a second. But another cool thing just about all invertebrate life in the ocean have incredibly impressive life histories, and sunflower sea stars are no exception. They spend the first month or many months in the plankton, so floating about after they've released, so this is an adult releasing eggs that we're collecting, but in the wild this would just be all wafting out into the ocean. There would be a male also releasing their gametes. They'd have fertilized eggs, and then slowly develop in the water column to this feeding version where they look like little spaceships and astronauts. So the red part and on the top corner ones are actually the food that they're eating. That's microalgae. So when they're in the plankton, they actually feed on those initial building blocks of life. And then right over here, these are very cool larvae. So this is when like the spaceship is ready to land. This is the larval body is really preparing this tip, which is called the rudiment. And this is actually the baby sea star. And it's going to use chemical cues in the environment to figure out when it wants to land on planet Earth. So come out of the plankton and go live the rest of its life roving the seafloor. And so this is a huge metamorphosis where this whole larval body gets reabsorbed into this rudiment, and they go from bilaterally symmetric, so like you and I, we have one side and the other that look alike, and then they go into this radial symmetry, which is also just wild to go through that type of metamorphosis. And they start with how many arms? 5. Yep, all sea stars start off life. This is 0.5 millimeter, so just barely visible to the naked eye, with 5 arms. And as they get bigger, you can see this is a little piece of a sea urchin test that was broken up that they're eating. And so this again is like a star that's less than the width of your thumb. And he's adding little arms at a time. And then as they get bigger, they look a little bit more like they're adults, and they grow into these beautiful sea stars. And here are just some beautiful pictures of Sea stars, in all the places you may find them. We love them in our kelp forest, but they're generalists in terms of where you find them. They can be found on mudflats and seagrass beds, in harbors, under piers, in kelp forests, and rocky reefs. And they're also generalist predators because they just want to eat whatever is easy and abundant. So if they're in intertidal habitat where they're prevalent mussel beds, so these are mussels that you'll see just if you're walking down the pier, or sea urchins, or even this is a picture of a wolf eel being eaten by a sea star. So whatever they can come across, they will try to eat. And so why do we care about sunflower sea stars? They are a keystone predator. They have the ability, in terms of their size, their relative speed, they're very fast for a marine invertebrate. I wouldn't go cheetah speed, but about this is about the speed of your doom to an urchin. This is really fast. And they like to eat purple urchins. So we have a great social media account if you haven't already followed Sunflower Star Lab on Facebook or Instagram, but we do like to have some fun with the fear that they can incite. So here's a video from our baby sea stars that were raised at the lab. Catching an urchin had no chance. This is actually the test of a urchin that was already eaten earlier that day. Okay, we'll watch that because why not? Yeah, just pounce. Okay, let's see if we can get away from here. There we go. Boop. And why are we excited about them eating purple urchins? Well, outside of this beautiful theater, we have the Monterey Kelp Forest, the Ed Ricketts National CMA, a protected area where we like to go diving in the kelp forests. And when they're healthy and lush, they are truly awe-inspiring place to live in. And the Kelp Cathedral, as we like to call it, is truly on stage today with the light beaming through those golden fronds. And because they're an algae, they also produce oxygen through photosynthesis, and that's not me. Okay, sorry. But also just their presence reduces wave action and reduces coastal erosion. They are habitats for thousands of species, from the whales that migrate through Monterey Bay, to the fish, to the invertebrates, to the jellyfish. And they are a carbon sink, whereby when they are broken off through storm waves, that their carbon is sequestered in the deep sea, like the trench just outside of our bay. But what happened to them? We had, in 2013, an unfortunate beginning of events. This was the historic range, all the hashed lines from the Aleutian Islands in Alaska, almost Russia's kind of right here, all the way down to Baja California. But the red is where they currently existed, or as of 2018, this was sort of the state of their population. So how did they get there? This is sea star wasting syndrome that was observed. This was an unfortunate turn of events that was noticed in early or late 2013, not just with sunflower sea stars, but with up to 20 different species were observed with this wasting event, where they would start deflating, losing their arms, and turning to goo. And within a day this would happen to individuals. And what was shocking is that just like humans and other animals, you can be, when you have a new disease sweeping through, it can be very hard to stop or understand what's going on. And the speed with which this sea star wasting event happened was literally within a blink of an eye. By, I think it was the summer in Washington State, it was first observed. And within a few months, it had ripped all the way down to California. I moved to California in LA in November 2013 and was doing surveys out at the Channel Island National Parks. And literally from one island to the next, you could see that the one island had wasting and the next one was just waiting to happen. So I came right as this staggering wasting event was happening. And several years after that, it made its way all the way up. So this image here, we show a lot. It is the most depressing slide, I think, of this whole talk, because this is the percent loss of stars. So you can see 100% loss in its southern portion of its range, and really greatly reduced numbers. So in total, about 95% of the stars, almost 6 billion stars, sunflower sea stars specifically, were lost in the span of 4 years. And this is what happened in Central California where we live. So Reef Check is a now region-wide surveying nonprofit where citizen scientists go into the ocean and take surveys, and we saw this huge decline of stars. Even if you didn't note the increase of stars prior, I mean, you went from about 0.1 stars per transect to 0 eventually by 2018. So for sunflower stars, those are the two other species that were recorded that also saw that loss. But then in the same respect, you saw the urchin populations increase. So the purple urchins and the red urchins that are found locally, one clearly took off more than the other, the gold with the purple urchins and the red urchins also increasing, but not to the same degree. So we had the sea star wasting event happen, and then in the following year was this giant blob that sat off the coast of North America and increased sea surface temperatures by almost 2.5 degrees. And it was tied with an El Nino that basically prolonged this warming through to 2016. And we saw a huge decline in kelp. This is just an image taken a few years apart, but and kelp comes and goes season to season with storms and everything, but the fact that the kelp hasn't been able to bounce back has been the largest issue. So the kelp was wiped out by these heat waves. They naturally die back in the heat, but now these urchin fronts who, they didn't have their predator to eat them, they had lost their own food because they eat kelp. It's great when you can get delivery pizza, but when the delivery pizza guy doesn't show up, you end up having to go to the store, find the pizza yourself. So these guys aren't the enemy, is what I'm trying to say. They're just really hungry and really good at eating everything. And they love kelp, and they'll literally climb up the stipes of the kelp and mow them off overnight in these urchin fronts. They just sort of all come up against the kelp forest and sort of unceremoniously take everything away. And that's not an issue just for the urchins, it's also a problem for all the other invertebrates that are grazing on the kelp. So red abalone, which is, of course, anyone was an abalone diver once upon a time, or knows family or friends that was an abalone diver? Yeah, so the fishery was closed in 2018 in part for red abalone because the kelp disappeared and all these abalone were starving along with the purple urchins. And we wanted to do something about it. There are a lot of wonderful initiatives that are based in Monterey and throughout the state that are trying to remove urchins manually with scuba divers, lacking any ecological solution, but we know that sunflower sea stars used to be here, and in order to maintain resilience with our ecosystems, and specifically the kelp forests, we want to help layer back some of those species and keystone predators that once roamed our kelp forest. So in 2021, we started, thanks to Vince Christian. Back in the day, he found out about a researcher up at Friday Harbor Labs that was raising sunflower sea stars to help understand that life history cycle that I showed you, those complex, metamorphoses and what they were eating and how you raise them. He'd finally published a paper on that in 2021, and Vince, coming out of COVID, drove all the way up to Washington to talk to Jason Hoden and ask, can we do what you're doing down in California, and do you know anyone who's doing it? And at the time, Jason didn't know. So Vince came down back to Monterey, posted this on Facebook, and asked if anyone would like to join him in learning about sunflower sea stars and saving them in Monterey. So in 2021, this was through social media. I was just finishing up my master's looking at the restoration of sunflower sea stars through public aquarium involvement in California. I've joined the board along with this wonderful group of inspiring community members that brought their expertise in aquaculture, in restoration, business savvy, nonprofit startup, and we started from there. But it all started in a little garage. But the whole reason why we had sunflower sea stars to begin with was because we partnered with public aquariums. And in 2024, we had a group of aquarists and research lab techs and our Sunflower Star Lab team go up to Friday Harbor Labs once we'd successfully spawned stars from the Birch Aquarium and learn about the care of sunflower sea stars. And this is all based on this manual that was published by the Nature Conservancy in line with the United Nations Decade on Ocean Science to ensure that we had a roadmap to recovery despite the lack of any state or federal recovery plan. Again, this species disappeared so very quickly, and there was definitely a lot of mobilized, inspired people that wanted to take action sooner rather than waiting around for a federal listing, which we're still waiting on, because you don't know what the government's going to do. So this was our first meeting in person of all these wonderful collaborators at the Monterey Bay Aquarium. This is on top of their Bechtel Center back in 2023. And I'm not going to go through this. We were checking boxes. This was a picture of the public aquariums and Sunflower Star Lab in California that came to the Birch Aquarium to share our spawning expertise and our ability to bring. This is the first time a female in California sunflower sea, the only confirmed sunflower sea star in California that's a female would have their spawn collected and then fertilized, and that's literally the first image that we had back in 2024 on Valentine's Day of a successful fertilization of sunflower sea stars in California. So that was a pretty big win. And we had lots of juveniles. We were very excited at the lab. We got to watch the whole life cycle unfold before our eyes. There's really nothing that you can do to replace having hands-on experience. And in 2024, later that year, we were one of the sub-awardees for a NOAA Pacific Coast Ocean Restoration Grant, along with 13 other sub-awardees. And we support our community because we came from the community and we're for the community. We want to reflect the community that is around us. And Minorities in Aquaculture is a fantastic nonprofit based on the East Coast, and we were their first West Coast host. And actually, Mariana is one of our full-time lab techs. She's phenomenal and came out of that internship program. And we, every day, all day, 7 days a week, are working towards improving conservation aquaculture, understanding the behavior, the welfare, the care of these animals. And it all started from the ground up. That was a first day we were handed the keys, and we slowly built all of this through our volunteers. And we've had some incredible photographers come to the lab and take some images that we can share with all of you and in our social media accounts and in just media in general, which has been great. And so this is, yeah. I mean, are they not spaceships or are they not spaceships? And you can really see, I mean, you can kind of see they're like little nubs. They're not arms yet, but they do kind of start having those sea star-like features, even when they're competent to settle. That's a technical term. When they're ready for landing, competent to settle. These guys are ready to go. They look beautiful. They've the little red arms, we call noodly arms. That's another super scientific term. They've got these long noodly arms that they can use to sense the environment. They're kind of like tacky at the end, so if they're holding on, they want to look for coralline algae, that pink rock-like algae that you'd see that coats the rocks of a rocky reef, or seagrass strands, and you'll actually see them sort of like tack onto it, and they have those suction cups here again. So on the top here, it's not in focus, but that little coloration is actually more suction cups, so it just helps them land and sort of secure themselves into that, ever-moving ocean. And then they take about a day or two to go through that metamorphosis. So they're vulnerable at that stage when they're putting all that effort into converting into a little baby sea star, like these guys. So this is just it's like a little, babies all grown up book. Look at them go. So and this is why you can't not call them fluffy. I mean, they're just so fluffy. And so this is all their papillae, so what they're breathing from, and then you can see the spines poking out through them, all those white parts. And the spines are actually really important when we're trying to identify them. So when we're thinking now about like restoration, how do you put a tag? You can't put a ear tag on like a cow. You can't put write something like you would on an abalone shell. They don't really have any hard features, and their soft features are so soft that, again, one of their great feats is that they can drop any arm. So if they have some foreign object in their arm, they'll usually just drop it. So you can't put a tag in them. If you dye them, it doesn't last very long. People have tried tattooing them. This has been a whole thing. So the one solution is, like we do with whales, is you take pictures of certain patterns. And what we found with the patterns is, even though they're very fluffy and they don't really have like a really nice flat surface, the spines are still somewhat uniquely spaced out by individual, that if you can take a picture with that mandriporate, that little button to anchor it, again for like machine learning or just for humans to sort of anchor that spot to get an orientation of, okay, like this is the upside of the picture, then you can somewhat tell them apart. We're still, there's a PhD student up at the University of Washington that's working on that. And here's some, they gracefully move around. And there we go. And so this is some of I was telling you about taking pictures. So this is That green outline is just a computer program that's trying to take the surface area of the stars, because it's hard to tell like growth when you're looking at mass, volume, arms. You know, if you just take one diameter, well, what if there's only 2 arms? You know, like it's been a lot of fun just trying to answer the really basic questions. We actually did studies on feeding them different things because we're like, well, do they have a preference of food because it makes them grow faster, or it's just really whatever is most prevalent? We fed them some frozen krill and urchins, and they grew at the same amount pretty much the whole time. So they love eating whatever is in front of them. They are not picky eaters. And we present at conferences. We collaborate with geneticists that have taken genetics from the stars that we have and are trying to look at the population disparities, if there are any or not. So these are points on the map where genetic samples have been taken before and after wasting. And Lauren Schiebelhet's trying to figure out, she's the geneticist, she's trying to figure out if there is any genetic distinction. And we are, the word's still out. Right now, they're all considered like one population. There's no subpopulation. And again, because I was telling you, those larvae, they float in the plankton for a month or more. They can stay and float, you know, down the current, down the coast of Oregon and go from Washington to California, in theory, just within that planktonic phase. So they're pretty well mixed up and down the coast from Alaska to Baja, as far as we know. And this is a lot on this one slide, but we've done a lot in a very few years, and this is not even up to date. But this all started back in 2021 when we were meeting at Vince's house, and we were able to collaborate with all of these wonderful researchers that bring their distinct expertise to answering the 1,000,001 questions needed to think about reintroduction and raising animals and caring for them. And here we go. We are the only Sunflower Sea Star dedicated institution on the West Coast, and we love partnering with lots of people. We believe in the restorative power of sunflower stars as an ecological solution to the kelp crisis and the overgrazing of urchins. And we are a volunteer-based board, and now we have 7 paid staff. And we love seeing from the community all your interest and enthusiasm, and it makes us want to show up for work every day. We do have a few volunteer positions open. They're none active right now, but of course, if you go out onto the coast of Northern California and you want to post on iNaturalist, we follow iNaturalist greatly. It helps inform where we want to take a restoration next steps, and you can participate by just posting what you see. And even if you don't see a sea star, that's actually good information too. So thank you so much for today, and I'll take your questions. Yes? For your guys' plumbing for in the lab, is it open or closed? We're recirculating, so we are not open. We take water from Moss Landing Marine Labs, which we love them, but we're not. A lot of people think that we are Sunflower, or we are Moss Landing Marine Labs, but we are a separate entity. But we do take their water, and then we have full life support system to help maintain good water quality. Yes. Yeah, we are, again, there are disease ecologists that have been working on identifying the pathogen, and this year they published a paper identifying a Vibrio species, Vibrio pectinacida, that was the main associated bacteria with wasting, and they did a bunch of different experiments to make sure that, this was clearly a causing agent of wasting by doing blind tests, inoculating them with 6 stars coalomic fluid, that the fluid that drives this whole species. And what we're doing here in Monterey is, because we know that this Vibrio exists, all the work was done in Hakai and BC, so in the inland waters. We want to make sure that we are monitoring that here and trying to figure out what's wasting look like here and what might be driving it in Monterey, because even though the wasting event was staggering and ecosystem altering, it still exists in small amounts. Like, we notice wasting pretty much up and down the coast every year in multiple species to this day. Just like when a new flu comes out, everyone it wipes out a lot of people, and then we have to deal with it forevermore. Wasting events have been observed in over history and time and space around the globe with sea stars, but never to that particular event. So we're trying to understand what's the threshold. What are sea stars that are still thriving in Monterey looking like when we have been working with the Department of Fish and Wildlife to get permits to take coalomic fluid from the sea stars here. Over the years now, we want to start this monitoring to sort of understand if there's any seasonality, where is the disease just hanging out but not showing signs of wasting, because where does the bacteria just hang out when it's not terrorizing everything? We're trying to figure that out, and that's we know we have a name, but that's just the beginning of, trying to understand what's going on. So we have a lot of work cut out for us. Yeah. And within the lab, we make sure that we UV the water, and they're filtered down to micron size that will limit the amount of bacterial buildup in our lab. So at least from the care within our lab, we are quarantining.
So have you started out practicing and
Wow. Do you have 5 hours? Honestly, the permitting has always been a thing with all restoration efforts, and we're very intentional right at the very beginning to bring in our permittees to the process. In California, I mentioned the Cupid cohort is what we call the stars that were produced from that 2024 spawn at Birch Aquarium, but it was 1 male and 1 female. So every single juvenile that are 2 years old right now in California are all full siblings. They're all brothers and sisters. That, in anyone's mind, should not sound like the way that you're going to restore a species with just adding full siblings into an area where there's no other sea stars to breed with. So this was really just a proof of concept, a proof to the state, a proof to all the institutions in California that we have the ways and means and the capacity, and with the funding that we've had from PCOR and institutional fundraising, that there's a need and a want to do this. And now with the California Department of Fish and Wildlife, and with all of our coastal partners, like our folks in Monterey Bay and everyone up and down the North Coast that share the same concern about wanting to rebuild our failed ecosystems off of our coast, we're being very intentional about how we go about building that up. And so we haven't released animals into the wild, but we have started doing ocean experiments. So last summer we were permitted by the department to put stars out in cages and see how they do, essentially. There were 2 reasons why we did that experiment, that we asked for this permit. One, a lot of people were wondering, if you have all these stars that are in a cushy little, beautiful, happy, healthy quarantine system, and then you put them into the ocean with everything living in it, how are they going to do? So with our partners at the California Academy of Sciences and the Sunflower Star Lab, we took 24 stars from each of those labs. Our lab had seawater that was being cleaned and treated, and then Cal Academy of Sciences has 100% artificial seawater that they raise their stars in, and we put them into Camp Ocean for a month and let them see how they did. So we took coelomic fluid, we did behavioral studies, so like their writing moment. A way to tell how healthy a sea star is you turn it upside down and see how quickly it takes to write again, just as a normal behavior. No one wants to be exposed on their back, and so we did that before we put them out into the ocean, and took coelomic fluid samples. So we took the liquid from within the star, sent them off to get tested to see if they had Vibrio. They didn't, and then we put them in the ocean, let them hang out. We fed them, measured their growth. They grew, and then took them back and took the coelomic fluid again to see if they picked up. What did they pick up? How did their microbiome change while they were hanging out in the ocean? And quarantined them, and they've been with us ever since. That was in October. So that was a big win. That was a small step, but a huge, again, show that not only were they healthy, but that we had the support from our local community members to execute that field work. Because if you were just to release stars, they can go and find whatever food is out in the ocean. But when you have to keep them in a cage, they're basically under human care still. So we had to go out twice a week, feed them. We did behavioral assessments every time we went out there. We had temperature and pH meters out there to understand sort of what their water quality was like. And we really basically just put them out in camp. And now that's sort of helping us build more questions that we can ask without releasing them for this year. And on top of that, we were able to do an environmental DNA study. So this is a whole other talk I did not talk about because I could go on forever. But environmental DNA is, all species like you and I, as well as every fish and invertebrate in the ocean, whether you're a larval animal or a huge sea star, will shed DNA because you're constantly creating cells and shedding that into the environment. And we can take a scoop of water, put it through a filter, do some fancy science, and identify the species that are in the ocean. So one of the things we had a grant for was to develop an assay that would just identify and pull a unique sequence of DNA, so like the ATCGA, the building blocks of life, that's unique to this species, and try to do a field experiment where we had, since the stars were in one spot in the ocean, we had free divers come and take water samples at increasingly further spots radiating away from this one cage of sea stars to see how sensitive this tool was to identify the presence or absence of sea stars. Because if you can't imagine, trying to understand the population change over time of sunflower stars moving forward when they're really rare, even though every diver would love to search day and night for sea stars in the ocean, it's not that practical. And if we can take a water sample and detect a sea star in an area that they haven't been seen in 5 years, that's great. That's when we can say, okay, we took a water sample in Santa Cruz and we got a hit in this bay. So now let's send our scuba divers out to do a search to see if we can find them. And so we used the fact that we were going to put stars in the ocean for this field experiment to test that tool, so that water sampling tool where we can sort of detect sea stars from water. Yep.
So when they come, you can start
If you know, you tell me. But that is clear. That is the question of the day. Is you know, how much effort do you have to put into caring for them before you put them out? Again, these are broadcast spawners. They literally produce millions of offspring with the knowledge that only 0.1% is going to survive. So what's the best way to go about, allowing that natural selection to occur? These are all questions that, as a huge collective, we are figuring out. And probably what the next steps would be to, and this is happening in Oregon and Washington already, because they can translocate animals because they have sea stars sunflower sea stars that exist in Oregon and Washington, and they can start doing those experiments of like what size and how many, and we're getting there in California, and we're really excited to be part of that. So,
Thank you so much. Thank you.